Hello, in this lecture, we will do a short introduction to stem cells. Specifically, we will discuss different types of stem cells and some of the applications for stem cells in culture research. Stem cells are the parental cells that give rise to every different type of cell in a multicellular organism. As you can see in this figure on the right, stem cells can differentiate into any type of cell, such as a red or white blood cell, a platelet, a nerve cell, or a liver cell. Stem cells are the progenitor cells, and they live, if you will, at the top of the totem pole. And differentiated cells are cells that have come down the totem pole and have one specialized function. Specialized cells cannot climb back to the totem pole. There are three common characteristics that define a stem cell. One, they are capable of dividing and replacing or renewing themselves, and somatic cells are not. And this can happen for a long, unlimited period. They are unspecialized to some degree, meaning they don't have a specific function, as they are progenitor cells that can give rise to specialized cells. That's point number 3, they give rise to more specialized cells, as well as renewing their own unspecialized type. Here's a simple but fun explanation of stem cells. It is geared for kids, but hey, we're all kids at heart, right? Some of us more than others, so I hope you enjoy this. Stem cells can be further divided into different types, depending on their stage of development. Embryonic stem cells come from embryos, and we call those totipotent, as totipotent cells give rise to all cells in the organism. Non-embryonic or adult stem cells, we call those multi or unipotent. Induced pluripotent stem cells are adult stem cells, but that have been laboratory manipulated. Cord blood stem cells or amniotic fluid stem cells, those we call multipotent, and hematopoietic stem cells are stem cells that give rise to different types of blood cell types. There is difference between these types of stem cells. Here on the right table, you can see their potential. Totipotent cells can develop an entire new individual. You may find these 1 to 2 days in 1 to 2 to 3 days embryos after conception, so early embryos. Pluripotent stem cells are more specialized than totipotent. Totipotent stem cells are completely unspecialized cells. These pluripotent can range anywhere from over 200 cell types, and you can find these in blastocysts about 5 to 14 days after conception. Multipotent stem cells are somewhat differentiated, but can still form a number of other tissues, such as fetal tissue, cord blood, or even adult stem cells. In research, we have most access to both pluripotent and multipotent cells. And in this table, we can see some of the advantages and disadvantages of both. Pluripotent stem cells can be advantageous in that they can develop into any specialized cells. They are relatively abundant, and they are easy to identify in a laboratory. However, because they are derived from embryos and fetuses, these type of stem cells are somewhat controversial. Also, there is always a chance of immune rejection if used in a patient. Multipotent stem cells are advantageous in that they can be isolated from adult patients, and there is no chance of immune ejection if it comes from the same individual. Some of their disadvantage is that we can only find multipotent stem cells in certain tissues. They are difficult to find and isolate, and they are not in abundance. There are two fundamental properties of stem cells that relate to their long-term self-renewal that scientists are trying to understand. One, why can embryonic stem cells proliferate or divide for a year or more in the laboratory without differentiating, without becoming a specialized cell, but most adult stem cells cannot? And two, why are the factors in living organisms that normally regulate stem cell proliferation and self-renewal? One of the biggest hurdles in stem cell research has been to keep the cells from becoming undifferentiated or specialized cells. It took 2 decades to grow human embryonic stem cells in the undifferentiated state in culture after successfully culturing mouse embryonic stem cells. Semi-successful in culturing adult stem cells has been achieved, but more work is needed to get larger numbers. Keeping cells in undifferentiated state requires very specific culture conditions, very stringent conditions, more than you have seen in our lab. There are signals that cells recognize as differentiation signals, and these include chemicals that are secreted by other cells, physical contact with neighboring cells, so how confluent the cells become. Certain molecules in the microenvironment can cause them to differentiate. The combination of these signals during differentiation causes epigenetic marks or changes in the way the DNA is regulated, and this will restrict DNA expression in the cell. This can then be passed on to daughter cells through cell division and can lead to cell differentiation.
So what are the uses for stem cells? Well, one of them is medical conditions, specifically for diseases such as cancer or birth defects, which are due to abnormal cell division and differentiation. A more complete understanding of genetic and molecular controls of these processes may yield information about how such diseases arise and suggest new strategies for therapy. The promise of stem cells is cell-based therapy, or replacement of damaged or non-functioning tissues and organs. However, more precise drug screening is necessary. We must get better at consistently producing the cell types of interest, and we must also get better at organ regeneration, which is still very untested except for skin replacement. So what are the requirements for stem cell culture? To be useful for transplant purposes, stem cells must reproducibly 1. Proliferate extensively and generate sufficient numbers of cells to make a tissue. 2. Differentiate into the desired cell type. 3. Survive in the recipient after the transplant. 4. Integrate into the surrounding tissue after the transplant. 5. Function appropriately for the duration of the recipient's life. And 6. Avoid harming the recipient in any way. There's still much work to realize this potential. Stem cells have much potential for future therapies, but we still have mountains to cross. There's still significant technical hurdles to subdue. Much research is still needed and required. Here below, you can find some federal and state agencies coordinating and funding stem cell research. I hope that you have found this short introduction